Good evening, everyone. Welcome to Botany Insiders second day revision series. So, please confirm whether the audio and the video is clear to you or not. Good evening, Mayank. Others also, please confirm whether the audio and the video is clear to you or not. Let's wait for students to join in and then we'll begin with our class, okay? So, I hope this particular segment of revision series is helping you out. Yesterday, we had a discussion regarding that what chemical bonds are, what are its various types, right? And today we'll be discussing regarding what nucleic acids are, what are its type, what are the various functionalities depending upon the importance it holds in the CSIR net and the other examinations that are coming up, right? So good evening Nisha, good evening Khushi. Let's wait for everyone to join in, then we'll begin with our class, okay? Just one more minute and then we'll begin. It's a very important topic. If you look into the CSIR's examination, like previous year questions and other examinations also, this particular topic holds a very important place, okay? So let's begin with our class now. First of all, if you're new to Botany Insider, make sure you hit the subscribe button. Make sure you visit our application via downloading it from the Android as well as iOS, available for both the users. So download the application, just write down Botany Insider either on Android or iOS. You'll be easily able to download the same, okay? So this is about it. Then talking about the new course launch, so we are about to launch a new course on 4th of April. So those of you who wish to get into these practice sessions, it starts on 4th of April, live sessions where we'll be discussing previous year questions, all the previous year questions for you all to understand, to get to have a complete guide regarding कि questions को कैसे solve out करते हैं exams के अंदर, क्या क्या tricks हमें use करनी चाहिए, किस तरीके से हम best answer को predict कर सकते हैं. So this particular course will help you out in the same. In addition, you can download the app as well as give your place your orders for the revision booklets and the PYQ pointers via WhatsApp or this via WhatsApping on this particular number. There's a combo offer going for you all. So whatever you wish to have, you can download the app and get yourself the required content, okay? This was about it. Let's begin with what nucleic acids are all about. See, अगर हम previous year questions की बात करें या फिर अगर हम न्यूक्लिक एसिड टॉपिक के इम्पोर्टेंस की बात करें, आप प्रीवियस ईयर क्वेश्चंस में देखेंगे बहुत बार दिस पर्टिकुलर क्वेश्चंस गेट रिपीटेड कि न्यूक्लिक एसिड का कॉम्पोजिशन क्या है, या फिर न्यूक्लिक एसिड के अंदर मेजरली दो टाइप के आते हैं, वन इज डीएनए, वन इज आरएनए. सो डीएनए एंड आरएनए में डिफरेंस क्या होता है, फर्स्ट थिंग. सेकेंड थिंग, दोनों का स्ट्रक्चर क्या है, दोनों की इम्पोर्टेंस क्या है, एक्सेट्रा एक्सेट्रा. बिफोर अंडरस्टैंडिंग कि डीएनए और आरएनए होता क्या है, हम पहले न्यूक्लिक एसिड एज अ होल की कॉम्पोजिशन को समझेंगे. सो एक न्यूक्लिक एसिड के अंदर हमारे बहुत सारे न्यूक्लियोटाइड्स बहुत सारे बॉन्ड्स के थ्रू अटैच्ड होते हैं, राइट? सो दिस अटैचमेंट हेल्प्स इन व्हाट? दिस अटैचमेंट हेल्प्स इन the linking of these nucleotides very nicely, and as a result of the same हमारा एक stable nucleic acid का formation होता है. So nucleic acid is made up of nucleotides, और अगर हम nucleotides के, you're not able to hear my voice, is the same problem for everyone or just for one student? Just check, because I guess from my end it's working absolutely fine. Now check. Hello, mic testing. Hello, is it working fine now? Other students also, please confirm it in the chat section. It's clear for you. So other students, if it is causing any problem, please check it from your end, because अगर मेरे एंड से प्रॉब्लम होगा तो सारे स्टूडेंट्स को प्रॉब्लम आएगा, बट अगर कुछ बच्चों के लिए ठीक है, सो आप अपने एंड पर एक बार ट्राई कीजिए. Fine, now I have not done any change, it's absolutely the same. Okay, doesn't matter. So nucleic acid के अंदर majorly two types है, DNA and RNA, right? Nucleic acid जो है ये बहुत सारे न्यूक्लियोटाइड्स का एक कंप्लीट लीनियर चेन है. सो जब बहुत सारे न्यूक्लियोटाइड्स एक स्पेसिफिक फैशन के अंदर एक दूसरे से कनेक्टेड होते हैं, वी कॉल इट एज अ न्यूक्लिक एसिड. न्यूक्लियोटाइड्स के अंदर दो सब कंपोनेंट्स हैं. The first one is nucleosides and the second one is phosphate group. So nucleosides and phosphate group. Nucleoside के अंदर again, okay, I'll use English also. Actually some students want me to use English, some wants me to use Hindi, some want to have bilingual. So I cannot use all the three things all together, so it's a bit difficult for me to accommodate this thing. You all decide yourself which type of language you want, and then only I can proceed further, because I cannot take up everyone's choice and then proceed. That is not possible for me, because some are saying that ma'am please use Hindi, I'm using Hindi, then it comes English, then it comes bilingual. So it's not possible for me to go for every type of request. Okay, so please try to understand my situation also, right? So nucleosides के अंदर and हमारे पास again there are two components. First one is nitrogenous bases, second one is sugars. So nucleosides have nitrogenous bases plus sugars. Nitrogenous bases के फिर further दो sub components हैं. First is purines and the second one are pyrimidines. Very important, purines and pyrimidines, both of them are what? Purines and pyrimidines. Again there are various types of purines and pyrimidines. Now in the examination the biosynthesis of purines has been asked directly, biosynthesis of pyrimidines are also asked directly. So you need to make sure that you are keeping a good hold over these topics, right? Purines के हमारे पास दो types के purine हैं, या फिर दो nitrogenous bases, that is adenines and guanines are under the subclass of purines. And pyrimidines के अंदर हमारे पास तीन सब classes हैं, cytosine, uracil and thymine. I have tried to make this particular diagram, this complete flowchart as simple as possible. It won't cause any kind of confusion to you, so try to have a very good grip over the complete composition of the nucleic acid, right? Then we have the sugars. In the sugars you have deoxyribose and the oxyribose, right? deoxyribose and oxyribose के basis पर ही हमारा DNA और RNA का distinguishing होता है. Like how do we actually distinguish between a DNA and a RNA? It is dependent upon the type of sugars present. When you look into the full form of DNA, it says deoxyribose nucleic acid. So deoxyribose sugar हमारे DNA में है, और RNA के अंदर you have ribose sugar. Okay, so dependent upon the sugars type, we have the type of the nucleic acid. In addition to the distinguishing in the type of sugars, we also have the difference in the DNA and RNA dependent upon what type of purines and pyrimidines are present, right? So in the purines or in the case of DNA, what do you have? You have A, T, G and C, that is adenine, thymine, uracil, guanine and cytosine. In case of ribose or the RNA, you have all of them similar. In place of A, in place of T, you have U being there, that is adenine, guanine, uracil and cytosine. So uracil is the same one. Umesh, if there's a problem in voice, I guess it's from your end, because for other students is working fine. Okay, so please check it from your end. If there's a problem, I cannot help out in this thing, because for all the other students the voice is working fine. Please try to increase the volume from your end. dideoxyribonucleotide, how to remove 3' O group. Means I guess we are studying about the nucleic acid, so I am going only for the basics right now. So please concentrate upon whatever I am teaching. We'll take up the questions at the end of the class or in the class which is for the doubt session specifically. So please concentrate upon the topic that I am teaching right now. Okay? Sometimes it's getting very low. What is the problem over here? Let me check. It's working absolutely fine from my end. Maybe because of the weather, because the weather is not proper in my end. Let me check. Hold on a second. Okay, I guess it's working fine now, right? So you have the nucleic acid. This is made up of a number of nucleotides. So polynucleotides. In the nucleotides, you have nucleosides and the phosphate group being there, right? In the nucleosides, we have nitrogenous bases and the sugars. Nitrogenous bases are of two types: purines and pyrimidines. Purines, adenine and guanine. Pyrimidines, cytosine, uracil and thymine. Then in the case of sugars, you have deoxyribose and ribose, right? Just check, is everything clear to you till here or not? If yes, then we'll proceed further to other points, right? Quickly check, is everything clear to you or not till this particular point? Clear everyone till here? Okay, let's proceed further now. Right now you have three parts in a nucleotide. What are the three parts? First one is a phosphate group, then you have the pentose sugar, then you have the nitrogenous base over there, right? So phosphate group, pentose sugar and nitrogenous base. Depending upon the difference in the nitrogenous base and depending upon the sugar that is being there, there is a change in the type of the nucleotide, right? Either DNA or RNA. So next, you have the biosynthesis of purines and the biosynthesis of pyrimidines. So see, I have tried to keep it very, very simple. This is from my own notes. What is given to you over here? So in the pyrimidine's nucleus, you need to keep in mind that which particular element is getting, or what is the source of the particular element. Okay, so N1, sixth carbon, fifth carbon and fourth carbon. That is, when you look into a pyrimidine, in a pyrimidine there are in total 6 elements, right? And there must be some source wherever it is, from wherever we are getting it, and finally this particular structure gets formed, right? So you need to keep in mind, you need to make sure that you are remembering all the sources. So N1, like the first, at the first position the nitrogen, and the carbon at fourth, fifth and sixth position, all these are derived from aspartate. Okay, then you have the second carbon getting derived from CO2, that is, and this carbon dioxide is via carbonyl phosphate, right? Then you have the third nitrogen. So this third nitrogen is derived from the glutamine. So in the pyrimidine's structure, remember the basic functionality, remember the basic source of each of the element, that is first, sixth, fifth and fourth, all these are via aspartate, second carbon is via CO2, and the third nitrogen is via the glutamine. And in the examination, direct straightforward question has been asked, like exact same thing was asked in the examination regarding the source of each of the element in the pyrimidine and in the purine as well. If you look into the previous year questions, you'll get to understand that these type of things are asked in the examinations very very often. So please make sure that you are keeping a note of this particular thing, right? In addition to that, you have the biosynthesis of purine. So in the biosynthesis of purine, it is a double ringed structure, and in this double ringed you have, and it is also 9 membered, okay? And in these 9 membered, what you have to keep in mind, N1 is via aspartate, second carbon is via N10 formyl tetrahydrofluorate, second carbon and the 8th carbon are via the same sources, then third and ninth carbon is via glutamine, fourth, fifth and seventh elements are via glycine, sixth is via carbon dioxide, and yeah, so remember all these things. Direct straightforward thing was asked, so I've kept a note of this. So you need not to go into the depth of how is it actually formed. If you keep in mind, if you keep this complete information to yourself, it would easily help you out to solve out the answers asked in the examination. So I've kept a list over here as well. You can keep a note of this. In addition to that, you can keep a note of this as well. You can take a screenshot from here and get your like in your notes you can add on this particular information. So biosynthesis of purine and pyrimidine, both of things are added over here. Quickly check whether this particular thing is clear to you or not. If yes, then we'll proceed further, okay? Clear? Very good. Let's move forward now. Now let's understand about DNA, right? So what is DNA all about? So DNA is the main principle DNA is the main principal genetic material from the bacteria to the humans. It runs in a opposite direction, that is in the antiparallel nature. Now when we look into the DNA and the RNA, let's try to understand few more points. Yeah, so when you look into the nucleic acid, majorly we have first thing as the DNA and the second thing as the RNA. So in the DNA, which are the nitrogenous bases that we have? So in the DNA there are 4 nitrogenous bases, where you have adenine, then you have thymine, then you have guanine, then you have cytosine, then you have cytosine, right? In case of RNA, all the 4 are similar, all the 3 are similar. The major difference is with the presence of thymine. So in case of RNA, you have A, U, G and C, that is adenine, uracil, guanine and cytosine, right? So remember this main distinction, that is DNA is ATGC, RNA is AUGC, okay? And when we look into the hydrogen bonds which are formed, so in case of DNA, A forms double bond with T, so adenine and thymine would be forming double bonds with each other and stabilizing the structure. Then guanine and cytosine, they'll be having 3 hydrogen bonds in between each other and then forming the bonds, right? In case of RNA, you have A forming double bond with U and G forming triple bond with C. The only difference that occurs is that in case of DNA there is T, in case of RNA there is U being there. The number of hydrogen bonds would remain always similar. So AT 2 bonds, G and C 3 bonds. In case of RNA, A and U 2 bonds, G and C 3 bonds, right? So these are the number of hydrogen bonds. Third important thing is the presence of OH at the second and the third carbon. So when it is deoxyribose, so there is a absence of an OH group at the second position, and in case of RNA there is an additional OH group present at the 2' site. So because of the presence of an additional OH in the RNA bonding site means I have not added that particular structure. Tomorrow also, can we use Chargaff rule for RNA? Chargaff rule actually describes about the number of ATGC present. So that is, I don't think it can be applied for RNA. I have to read more about it. I'll let you know. And the other student, tomorrow also I'll be continuing the nucleic acid series itself. Tomorrow also I'll continue the same topic. So I'll add all these things in that particular class. Okay. So what we were discussing, yes, in the RNA there's a presence of additional OH, because of which the RNA is less stable. Reason being, it can easily get attacked by the nucleophile. So remember about this, that DNA is more stable and it is a more common nucleic acid. Reason being, it have an one less OH group being present, and as a result there is less chances of nucleophilic attack in the DNA structure. So the DNA is more stable as compared to the RNA. So these are the major distinctions that you have to remember between DNA and RNA. DNA, nitrogenous bases, then hydrogen bonds, then the presence or the absence of the OH is helping out in the formation or the increase or the decrease in the stability of the DNA and the RNA, right? There are various types. In addition to that, one more thing, the DNA is double stranded and the RNA is single stranded. Remember this particular thing as well. It is a double helical structure and the RNA is majorly single stranded, right? Next you have the, yeah, now I guess let me give you one question so that you can solve. Yes, depending upon whatever we have discussed, try to find out the answer for this particular question. The question is saying, on the sequence analysis of a double stranded DNA, result shows that the cytosine was present in 20%. Okay, then it was asking that what is the amount of A and T put together, right? So in this question itself you can apply your Chargaff rule and easily get the answer for this particular question. Okay, very simple question. It is a previous year question that was asked in the CSIR. So try to find the answer for this question. Is saying you have 20% cytosine being there, so it is asking what is the amount of A and T put together, right? Try to find the answer for this question, everyone. Give it a try. Umesh, if RNA is there in it is double stranded in some RNA or some viruses or bacteria, that doesn't mean we can apply a universal law to it. Exceptions are there in each and every condition that we study, right? So if and all there are exceptions, we cannot apply universal laws to exceptional cases. Remember this thing, not just for this particular thing, not just for this particular topic, but for other things also. Remember that if any particular entity is showing some kind of exceptions which is normally or common in nature, that does not mean you can apply a universal law to that. Okay, yeah, let's try to find the answer for this. So it is saying cytosine is 20%. So if cytosine is 20%, guanine will also be 20%, right? Because guanine and cytosine, they both form bond with each other, and they both will have to be in the equal concentration in order to maintain the proportionality in our structure, to have a what do you say, the constant or a very stable structure. For that you need to have a equal concentration of the bond of two nitrogenous bases forming together, right? So adenine sorry cytosine and uracil cytosine and guanine are having bonds with each other, and if one of them is 20, other will also be 20. So in a DNA you have four main nitrogenous bases, that is A, T, G and C. If G and C all together is 40, then A and T would be what? It would be 60, because in total there are four present, so it would be 100%. If out percent is occupied by G and C, you'll get the remaining the two as 60%, right? So answer is 60%. And I'm giving you one additional question over here. What if the question was asking that what is the percentage of adenine in the complete structure? Then what could have been the answer? Think about it, everyone, and mark the answer. Yes. If the question was asking that adenine percentage would be what? 30%. Yes, remember this particular fact, that if adenine was asked, so adenine and thymine, both of them together is 60, so both of them would be in equal concentration itself, so it would be 30% for adenine or for the thymine, whichever is asked in the examination, right? So this was about it. Tomorrow also I'll continue the same series. We'll discuss few questions also for nucleic acid. In addition, we'll try to have the various types of the DNAs, that is A form, B form, Z form. We'll try to have the distinguishing, like the differences between all of them, discussed in tomorrow's class, right? So please let me know whether you are liking this particular format of questions or not, and this complete format of revision sessions or not, so that we can decide accordingly, should we continue this series or should we try out any other format. Yes, is this particular series helpful for you, everyone? Molecular genetics kara do. I have kept a number of topics for this particular thing, so I'll let you know. For the complete like revision sessions, we have a number of topics enlisted, so I'll go one by one. इस टॉपिक के बाद डेफ बायो करा देना. डेफ बायो को 30 मिनट्स के अंदर समराइज करना निशा पॉसिबल नहीं होता, बिकॉज इट्स अ वेरी लाइक इन लार्ज वेरी what do you say elaborative topics है वहाँ पर. Still I'll keep in mind about this particular thing. What is the percentage of hypoxanthine xanthine 5 methyl cytosine? I have to read about it, Santkil. I will read about it and then only I can answer that. Okay, I'll answer to you in the next class. For this I have to read about it. Dev bio. Okay, let me see if many students are asking for it. Let me see which topics we can incorporate in the revision sessions. Tomorrow we'll continue with the nucleic acid itself, and then let me see how we can incorporate dev bio over here. Okay, so for that you have to do, like from your end, you have to make sure that you are sharing about our channel to a number of students. Let's see how many students you all can bring in on our platform, on our YouTube channel, on our application, because we need students for me to conduct classes, right? So make sure that you're sharing about our channel, sharing about our application to a number of students, and let them come to our platform and practice prepare with us, right? So this was about for today's class. Again, a reminder, practice sessions are starting from 4th of April 2023. Those of you who wish to enroll, you can download the application and enroll in this particular course. We also have a combo offer running for you all for the revision booklets and the PYQ pointers for the June 2023 examinations, right? If you wish to place your order, you can WhatsApp on this particular number and get the sample PDF for the same and try out the same thing. Okay, you're sharing with Tamil Nadu friends, that's a very good thing. Thank you so much. Okay, so this was about for today's class. Thank you. Bye everyone, take care.